So let's go ahead and take a quick look at the material for Chapter 2. I would imagine the majority of this is going to end up being a review from your basic bio and basic chem classes. So elements such as carbon, hydrogen, oxygen, nitrogen, these are going to be pure and they cannot be broken down. If you take a look at these elements over here, these are going to be the elements that we will deal with the majority of our time in biology. Remember that you can combine elements to form molecules, which are going to be two or more atoms covalently bonded, such as this diatomic molecule of oxygen. And remember that atoms make up elements. All right, so we can combine atoms to form compounds, which are again two or more different elements in a fixed ratio. For example, the compound water, H2O, has 2 hydrogens for every oxygen. Remember that atoms contain a nucleus in the center and an electron cloud, which is going to surround the nucleus. In the nucleus, we're going to have our protons and our neutrons, which are going to equal our atomic mass. Neutrons have a neutral charge, and protons are positive, and they're going to equal the atomic number. So, for example, carbon has 6 protons and 6 neutrons, so its atomic mass would be 12, but because it has 6 protons, its atomic number is also 12. Now, the number of neutrons can vary from one form of an atom to another, and this would be called an isotope. So, for example, we have carbon-12, which would be the most common isotope of carbon, 6 protons and 6 neutrons. However, we also have carbon-13 and carbon-14. Carbon-14 is going to have 6 protons and 8 neutrons. So back to our electron cloud. The electron cloud, the outer shell is going to be containing our valence electrons. These are going to be the electrons that cause the atoms to have the particular properties that they exhibit. And remember that electrons are negative. So protons are positive, neutrons are neutral, and electrons are negative. All right, so again, as I was mentioning, the electron cloud is going to be involved in chemical bonding, and we have 2 types of bonding that we're going to speak of. At the moment, we have covalent bonding and ionic bonding. All right, so covalent bonding is going to share electrons, and ionic bonding is going to exchange electrons, and this is going to be because of the attraction between oppositely charged ions. We have anions, which are going to be negative, and cations, which are going to be positive. Now taking a look at the covalent bonding. In covalent bonding, again, we're going to be sharing electrons, and we can have single covalent bonds, double covalent bonds, and triple covalent bonds, and they will share pairs of electrons. 1 pair of electrons for single, 2 pair of electrons for double, and 3 pair of electrons for triple bonds. Okay, so different types of covalent bonds. We have polar covalent bonds and non-polar covalent bonds. Okay, so in non-polar covalent bonds, like hydrogen, oxygen, and carbon dioxide, we're going to have an even sharing of electrons, so there won't be a charge on either end of the molecule. But for polar covalent bonds, like water, okay, we're going to end up getting opposite charges. So on one end of a water, the hydrogen, you're going to have a positive charge, and then on the opposite end, the oxygen, you're going to have a negative charge. And this is due to the fact that oxygen is going to be highly electronegative. It's going to hold on to its electrons more often. All right, and this polarity is going to lead to something called hydrogen bonding. Hydrogen bonding, remember, is super important in the chemical properties of water, things like cohesion and adhesion, capillary action, specific heat, things of that nature. We have other types of weak chemical bonds. We have things called intermolecular forces. These are going to be things that help form 3D shapes of macromolecules. When we talk about proteins, we will talk about how the amino acid chains are going to have a specific shape, and that's going to be partially due to these intermolecular forces, disulfide bridges, hydrogen bonding, attraction between positive and negative amino acids. And then we also have van der Waals forces, And these are going to be fleeting, very brief attractions between positive and negative regions of specific molecules. All right, so this was just a brief overview of the material in Chapter 2. Again, I believe that the majority of this would be review for you guys. If you have any specific questions, please let me know in lecture.